Gene cloning. Complete explanation. What is gene cloning? Gene cloning is a molecular biology technique used to produce identical copies of a specific gene or DNA sequence. It involves inserting a gene of interest, GOI, into a cloning vector and introducing it into a host organism, typically a bacterium, where it can be replicated and sometimes expressed. Objectives of gene cloning: to amplify DNA fragments to study gene structure and function to produce proteins, e.g., insulin, growth hormones, to create genetically modified organisms, GMOs, for gene therapy and vaccine development. Steps in gene cloning process. 1. Isolation of gene of interest. The gene is isolated from genomic or complementary DNA using restriction enzymes, endonucleases. These enzymes cut DNA at specific recognition sites to yield sticky or blunt ends. 2. Selection of a suitable cloning vector. A vector is a DNA molecule used to carry foreign genetic material into a host. Good vectors must replicate independently, contain a multiple cloning site, MCS, have selectable markers, e.g., antibiotic resistance genes, common vectors, plasmids, bacteriophages, lambda-phage, cosmid BACs, bacterial artificial chromosomes, YACs, yeast artificial chromosomes. 3. Ligation of DNA into vector. The gene and vector are cut with the same restriction enzyme to ensure compatibility. DNA ligase enzyme is used to seal the gene into the plasmid, forming recombinant DNA. 4. Introduction into host organism. The recombinant plasmid is introduced into a host cell, commonly E. coli, using transformation techniques, heat shock electroporation, microinjection, gene gun, in plants. 5. Selection of transformed cells. Not all host cells will take up the recombinant plasmid, so selection is necessary. Antibiotic resistance genes, e.g.